great to finally meet you. I'm Sanderson. Would you like to hear about my research? I'm working to target a protein called MAX. MAX is a DNA-binding protein which can bind to other proteins, including MYC. MYC is the master regulator of broad cellular processes. It accumulates in promoter regions and activates pro-proliferative genes. MYC-driven tumors account for 28% of the 33 major cancer types. The protein structure of MYC is intrinsically disordered and thus has been considered undruggable. I am excited about MAX. It is important in transcription repression and activation. MAX forms transcription-terminating homodimers that can competitively outcompete MAX-MYC complexes involved in transcriptional activation. My guiding research goal is to confirm candidate small molecules which bind and affect the biological function of MAX, maybe inhibiting dimerization with MYC. MAX was genetically modified to incorporate a his tag. The subsequent purification process involved affinity chromatography utilizing a nickel NTA column, exploiting the affinity between the his tag on MAX and the nickel ions present in the column. The elevate proteins were ready for analysis of purity and concentration. SDS page assesses protein migration. The purification was successful as the protein was present at around 24 kilodaltons in both the eluate and concentrate. This is good for MAX. However, the concentrated sample was contaminated with 4 other proteins, as seen by 4 bands on the SDS page. The concentration of MAX6's his protein and its contaminants was determined using its absorbance at 562 nanometers and compared to a reference BCA protein assay. 299.5 micrograms per milliliter were purified. This includes the contaminants, but I am confident in this value as my reference curve have an R-squared value of 0.9975. Differential scanning flowmetry, DSF, is a technique which tracks fluorescence as a function of temperature, measuring protein unfolding. If MAX6's his binds to a small molecule, there should be a destabilizing or stabilizing effect, which would shift the TM50 value and measure 50% of proteins being denatured. We picked small molecules identified from a small molecule microarray, which included a nitrogen-nitrogen, carbon-carbon double binding to oxygen motif. They looked highly soluble with the potential of binding to the MAX residues important in MAX binding, arginine, asparagine, and glutamine. Identified 2 promising molecules with TM50s at 47.2 and 46.8 degrees Celsius, well over 2 degrees above the DMS at blank, meaning these are promising molecules that should be further tested with an EMSA experiment. An EMSA analysis, which assesses nucleic acid-protein interactions, will provide additional information about the way the small molecules bind to MAX. The next step would include a cell-based follow-up assay to further test the binding parameters of the small molecules. This would test the impact of the small molecules on cell proliferation, apoptosis, and other cellular processes affected by MAX and MYC in vitro. I'm very excited about the preliminary results from the DSF confirming 2 candidate small molecules which bind and affect the biological function of MAX, providing potential hope for many MYC-driven cancers.